Good afternoon, everybody. In this part, I want to talk about some selected tests on our blood. Before I start talking about these tests, I want to review the blood for you. You know that the blood is a liquid connective tissue. In this connective tissue, we have some formed elements like WBC or leukocyte or white blood cells, thrombocytes or platelet, and erythrocytes or red blood cells. The white blood cells can fight with different types of pathogens. Platelets can make blood clotting, and the erythrocyte can carry oxygen for us. And the liquid portion of the blood is called plasma. The majority of our plasma is water, but we have many dissolved protein, waste product, nutrient, gases, regulatory substances, electrolyte ions in our plasma. Last time I talked about the structure of these blood elements. I showed you platelets, which are the small fragments without any nucleus in our bloodstream, and they can stimulate blood clotting. We have leukocytes in different shapes and different sizes because we have different types of pathogens which invade our body, and we need different types of leukocytes to fight with them. And the erythrocyte is a biconcave disc with a very thin center. It's full of hemoglobin, and it needs to be smooth in the borders. And as you know, it can carry oxygen for us. The white blood cells are divided into two major groups, granulocytes and agranulocytes. In the granulocytes group, you can see neutrophil, eosinophil, and basophil. The neutrophil has up to five pieces for the nucleus. The granules in the cell are lilac in the color, which are full of enzymes and defensin, which can fight with different bacterial infections. The eosinophil has bilobed nucleus. It contains red granules, and it increases at the time of allergic reaction and parasitic worms. And the next one is called basophil. As you see here, in the basophil, we have many dark, large granules. They cover the nucleus and cytoplasm for us, and we cannot see the shape of the nucleus very clear. The granules of the basophil can release histamine and heparin. Histamine can make vasodilation, and it can trigger inflammation, and heparin is the anticoagulant factor. And both of these chemicals are released by our basophils. The agranulocytes are divided into two groups, lymphocytes and monocytes. As you see here in the lymphocytes, the ratio of the nucleus to the cytoplasm is very high. We have large nucleus and thin rim of cytoplasm. We have T and B lymphocyte. T lymphocyte can fight with the pathogen directly, while the B lymphocyte can make antibody, and the antibody can deactivate the antigen. And the last cell, which you can see here, is called monocyte. The monocytes can only circulate in our bloodstream for a very short time, and after 5 or 6 hours, they come out of the bloodstream, they go to connective tissue, and they become macrophage. They have kidney-shaped nucleus, and they are fighting with chronic bacterial infection. We have the other name for macrophages, which we call it antigen-presenting cell, or APC. I will talk about this process for you in the immune system completely. And we have different numbers for our white blood cells. Based on the abundance of white blood cells, we have a mnemonics: never let monkeys eat bananas. And as you know, N is the neutrophil, L is the lymphocyte, M is the monocyte, E is eosinophil, and B is basophil. Imagine that here we have a sample blood, and we count the number of each of these white blood cells here. If we want to have the differential count for each of these white blood cells, we need to add all of these numbers together to have the total number of white blood cells, and then divide each of these cells by the total and multiply it by 100. And then we need to check the percentages which we have with the normal value which you have in your lab manual, and tell me if they are normal or abnormal. If they are abnormal, what kinds of problems do we have? So please do it by yourself, and on the day of practice practical, I will review this chart for you. In this picture, you can see the lymphocyte, the neutrophil. This is again the neutrophil. Here you have basophil, monocyte, and eosinophil, based on the information which I gave you before. Look at the picture, and then I continue. And now I want to talk about clinically important blood tests. First, I want to start with hematocrit. You know that the percentage of red blood cell in the sample of blood is called hematocrit. And now I want to tell you how we can measure the hematocrit, this percentage. If we wrap the tourniquet around the arm, we draw the blood and place the blood in the test tube, and then centrifuge the blood components are divided from each other based on their weight. In the bottom of our test tube, we can see erythrocyte. In the top of our test tube, we can see plasma. And between plasma and erythrocyte, you can see a very thin film, which we call it buffy coat. This buffy coat is the aggregation of all types of white blood cells and platelets. Now, we want to work on the percentage of this hematocrit. Again, you can see the tube, which we call it capillary tube. If we centrifuge the capillary tube, you can see 3 parts in this tube. In the bottom of the tube, you can see red color, which represents the erythrocyte. In top of the tube, you can see a straw color liquid, which we call it plasma. And between them, you have a white film, which is the buffy coat. The other way is poking the tip of the finger to collect blood for hematocrit percentage. You can poke the tip of the finger with lancet, wipe off the first droplet of the blood, and you can put the capillary tube or hematocrit tube on the second droplet, and the blood comes up in your tube. Then you can centrifuge your tube and you can see the result like this. These results are from 2 different individuals, A and B. And you see that the height of red blood cells in A is different from B. And now we want to describe them. Imagine that this is the tube which you centrifuge it, and you want to calculate the percentage of hematocrit. You can use the regular ruler for these calculations, for this measurement. You can Measure the height of your red blood cell. First, imagine that you measure this height, and this height is about 3 centimeters. Then you measure the height of your total blood from the beginning of red blood cells to the end of plasma, the large bracket. And imagine that we measure it and it's about 7 centimeters. Now we can use this formula, the following formula, for calculation of our hematocrit. The hematocrit is equal to the height of packed red blood cell, which is 3 centimeters, over the height of total sample, which is 7 centimeters, times 100. And when we do this calculation, the result is 42.8%. This is the percentage of our hematocrit. And now I want to tell you how you can use this percentage. So the formula which we use for calculation of hematocrit is this. The hematocrit is equal to packed red blood cell height over the total sample height times 100. Now you do this equation and you have your answer. Your answer is 42.8%. Now how can you use it? In the male, the normal value for hematocrit is 42 to 56%. So if our sample is from the male, we are in the normal value. So if it's male, it's normal. And in the female, this percentage is 38 to 46%. So if the sample is from the female, again, it's normal. Pay attention, you need to memorize the normal value for male and female. In female, it's 38 to 46, and in male, it's 42 to 56%. In male, if the percentage of hematocrit in the sample of blood which you have is 41%, it's below than the normal value. So this person has anemia. But 41% in female is normal. If the percentage of red blood cells or hematocrit is above normal, we call it polycythemia. In the polycythemia, the blood becomes very viscous and it cannot flow in the blood vessels normally. If the percentage of red blood cell is below than normal, we call it anemia. In anemia, the blood can't carry enough oxygen. So the person is very fatigued, he can't or she can't do his task properly, and we have many problems. So when you have the percentage of hematocrit, you need to check it, compare it with the normal value, and you can make your decision if this person is normal or anemic or polycythemic. This is about measuring the hematocrit with ruler. But we have some cards which we call them hematocrit cards or hematocrit reader. You can see the picture of this card here. The card has many lines, as you see here, parallel lines. The first line is 0 and the last line is 100. When you centrifuge your capillary tube, you can roll this capillary tube on this card until the bottom of your red blood cell align and touch with 0 line and the top of your plasma placed on 100. And then you can read the number of your hematocrit. This number can automatically gives you percentage. Here we have 47, and when we want to read it, we call it 47%. In this picture, you can see we have a sample blood. We insert our capillary tube in this sample and the blood comes up. Then we seal the end of our capillary tube with the clay which we have in the lab. We centrifuge our blood and you can see the result like this. You have red blood cells in bottom and plasma on top. Then you start to roll over your capillary tube on the hematocrit reader. And when the beginning of your red blood cell is on 0 and the end of your plasma is on 100, you can easily give us the percentage of hematocrit. And then you can check if this percentage is in the normal value or not. This is how we can read the hematocrit by the card.
Let's take a look at the hematocrit results. The hematocrit tube has been centrifuged. We're now analyzing it with this card. In order to do that, I need to slide the tube left and right until the bottom of the blood is on the 0 line and the top of the blood is on the 100 line. Now we look right here at the top of the packed blood cells. That level of blood is laying against this dark blue line. If I follow it to the side, what I'm going to do is find a number. In this case, it's 55. So that means this sample of cow blood was 55%. That's the hematocrit reading.
So again, when you want to read the hematocrit by the capillary tube and the card, you can roll over and slide your hematocrit tube, capillary tube on your card. And when your 0 line is in the bottom of your tube and your 100 is at the top of your plasma, it means that you are in the good location. Then you can read the height of your red blood cells here, and it gives you the percentage. It doesn't matter if you read the numbers from the right side or from the left side. Both of them give you the same number. The next part is about blood typing. Nowadays, you may hear about finding your personality based on your blood type or blood type diets, or you can date a person based on the blood type, so you can be well matched with that person. But blood typing is much more than this. We have a table for our blood types donation and blood types receiving. And most students are afraid of this table. Don't memorize the table. I give you some tips to understand how you can donate blood and how you can receive blood. But the only thing which I encourage you is donation of the blood. If you can do blood donation, do that, because one blood donation can save up to 3 lives. In previous times, some physicians figure out that when they do blood transfusion, some patients die and have problems, very bad problems, and some patients can survive, and they don't know why first. Then they start to work on the blood cells, specifically the red blood cells, and they figure out that on the surface of our red blood cells, we have some markers which are genetically created, and they can make our blood types. Look at these donuts. In the first donut, you can see only blue sprinkle. If these blue sprinkles are called antigen A, the blood type of this person is A. In this donut, you can see only yellow sprinkle. Imagine that this is the red blood cell with antigen B. So the blood type of this person is B. In the third one, you have a donut with blue and yellow sprinkle. So if this is a red blood cell, you have A antigen and B antigen. So the blood type become AB. And the last donut is plain donut. You don't have any sprinkle on it. So we call it, if this is a red blood cell, without any antigen, we call it O group. So we have A, B, B, and O. And they are the glycolipids which we have on the surface of our red blood cells. Besides the ABO group, we have one RH factor, which is the membrane protein, and I talk about it later for you. Look at this picture. In this picture, you can see red blood cell with A antigen. The blood type in this person is A. In the next one, you can see red blood cell with B antigen. The blood type of this person is B. In the third one, you have A antigen and B antigen. So the blood type of this person is AB. And in the last one, you don't have any antigen on the surface of red blood cell. So the blood type become O. You know that the antigens can stimulate our immune system to make antibody. Imagine that we have chickenpox virus in our body. Our immune system starts to make antibody against these antigens. If the surface antigen is A, in our blood, we have anti-B antibody. So if B group come to our body, anti-B is opposite to B, and it can destroy the new red blood cells. In the person with B type, we have anti-A antibody. It means that if in this person we use blood type A and anti-A can fight against each other and destroy each other. In a person with AB, when we have A antigen and B antigen, we don't have any antibody in the bloodstream. And in the person with O type, when we don't have any A or any B antigen, then we have anti-A and anti-B in the blood. Antigens are called agglutinogen, and antibody has the name which we call it agglutinin. They are produced against antigen. And pay attention. For example, for chickenpox, we need to be exposed with the chickenpox virus. so our body can make antibody against chickenpox. But for the blood types, we don't need to have wrong exposure. Our body automatically make antibody. And because these antibodies are automatically made, we call them performed antibody. It takes about 6 months after birth to produce these antibodies in our blood stream. Now, please check this picture and I will continue in 1 minute. So if a person is A, it has, he or she has A antigen, and in the blood we have anti-B antibody. If a person is B, we have B antigen on the surface of red blood cell, and in the blood we have anti-A antibody. If a person is AB, we have A antigen and B antigen on the surface of red blood cell, and we don't have any antibody in the plasma. And if the person is O, we don't have any surface antigen, and we have anti-A and anti-B in the bloodstream. Now, what happens if we have mismatching? For example, here you can see red blood cell with B antigen, which the blood type is B here, and you use blood type A in this patient. You know that B and anti-B are opposite. They want to attach together and fight with each other and destroy each other. So first we have clumping or agglutination, then the red blood cells rupture and the hemoglobin is released in the bloodstream. And this hemoglobin cannot carry oxygen. It can go out of the body and it's excreted by the urine. And the first sign of this mismatching and wrong blood transfusion is red pee or red urine, which we call it hemolysis happen in our blood stream. This is a normal blood, and the blood with agglutination is like this. You see that the red blood cells and antibodies are attached together, make clumping, and then they can destroy each other. The other antigen which we have on the surface of red blood cell is called D antigen or Rh factor. We have Rh positive and Rh negative. It's very easy. If we have D antigen, we are Rh positive. If we don't have D antigen, we are Rh negative. And pay attention, in both Rh positive and Rh negative individuals, we don't have any antibody against Rh, unless the person with Rh negative is exposed to Rh positive. Imagine that we have the Rh negative person and we give him the blood with Rh positive. This Rh positive or D antigen go to the body of our patient, and the body of the patient start to make antibody against Rh. In the first blood transfusion, nothing happened. But in the second one, if this problem occur again, then we have agglutination and hemolysis. So pay attention. We don't have any antibody against Rh in the blood in Rh positive and Rh negative individuals. And when we have a wrong blood transfusion, when we give Rh positive to a person with Rh negative, at that time, the antibody is created in the body of recipient, and in the next wrong blood transfusion, the body can show reaction. This antibody is called non-performed antibody because it's not automatically made in the body. Here you can see the different blood types. If you don't have any antigen on the surface of red blood cell, you have O negative. If you only have Rh factor, it becomes O positive. If you have only A antigen, it's A negative. If you have A antigen and D antigen, it's A positive. If you only have B antigen, we call it B negative. If we have B antigen and Rh or D antigen, we call it B positive. If we have A and B antigen, it's AB negative. If we have A antigen, B antigen, and D antigen, the person is AB positive. This is how blood types work for us. Now, imagine that I have a sample of blood and I want to see and identify the blood type. Look at here. Here you can see a glass slide. Sorry. In this glass slide, we have 4 droplets of blood. We will not add anything on the first droplet, but we add anti-A in the second one, anti-B with the third one, and anti-D with the fourth one. We use separate toothstick and mix the blood with these different antibodies. And we wait for about 30 seconds to 1 minute to see the reaction. If we see reaction with anti-A, if we see this agglutination, it means that we have A antigen. A and anti-A are opposite. Yes, so they make clumping. So in the first one, we have A antigen. You don't have any reaction with the third one. So it means that you don't have any B antigen. And in the last one, again, you can see reaction. It means that you have D antigen or your Rh is positive. So in the first row, the blood type is A positive. For the next ones, they are the same. You see that we have we don't have any reaction with anti-A, but we have reaction with anti-B and anti-D. It means that we don't have A antigen, but we have B and D antigen. So our blood type is B positive. In the third row, you see agglutination with anti-A. It means that you have A antigen. Agglutination with anti-B, so you have B antigen. Agglutination with anti-D, so you are positive. The blood type become AB positive. And in the last row, you see that you don't have any agglutinations in these droplets. So it means that you don't have any antigen on the surface of the red blood cell, and the blood type is O negative. have the slide in your PowerPoint which gives you different blood types and how they are treated with anti-A, anti-B, and anti-D. Please go over all of them, and you see the blood types here. But please cover these blood types and identify the blood types for yourself. And let me know next time if you have any questions. I go over, for example, this one. In this one, you see that we have agglutination with anti-A and anti-B. So our blood type is AB, but we don't have agglutination with anti-D. So it means that our RH is negative. Here you see that you don't have agglutination with anti-A and anti-B. So you don't have A antigen and B antigen. So the blood type is O. But you have agglutination with anti-D. So the blood type is positive, O positive. Now I want to talk about blood donation and blood receiving. In this diagram, you can see the process of blood donation and blood receiving. Look at the arrows. I don't want you to memorize it. If you look at the arrows, you can understand it. The beginning of the arrow here means you can give blood to a person. And the arrowhead is receiving. Now, in this picture, you see that the O can give blood to O, to A, to AB, and to B. But the O can only receive blood from one arrowhead, and this person is only O. So the blood type O is the universal donor. Look at A. The A can give blood to A and AB, but the A can receive blood from two arrowheads. One of them is A, one of them is O. The B can give blood to B and AB, and the B can receive blood from O and B. The AB can only give blood to AB, but the AB can receive blood from four arrowheads, from AB, A, O, and B. So the AB is universal receiver. You need to draw these charts 3 times, 4 times, 10 times for yourself to fix it in your mind. It can help you to the process of blood donation and blood receiving. You only need to check the arrows. I want to give you one example, and then you can do the rest examples yourself. Imagine that I have blood type, I have a patient with blood type A, and I want to give this patient blood. Who can give this patient blood? Which blood types? I need to check the arrowheads. I see that from 2%, A can receive blood. One of them is A, the other one is O. And in the other example, I have A blood type, and I want to donate it to different individuals. Who can I donate to? I need to check the beginning of the arrows. I can donate A to A and to AB. So by this method, you can easily understand who you can donate blood to and who you can receive blood from. In the next slide, it talks about RH type, RH positive and RH negative. Look at here, the RH negative can give blood to RH negative and RH positive. But the RH negative can only receive blood from RH negative. The positive can receive blood from positive and negative, but it can only donate blood to positive. Now, I want to give you the other example. Imagine that I have A negative or B negative. Blood, and I want to see who I can donate to. The B group can donate blood to B and AB. The negative can give blood to negative and positive. So if I have blood type B negative in the blood bank, I can give this blood to 4 blood types, B negative and B positive, AB negative and AB positive. Now, if I want to receive blood, what should I do? Imagine that my blood type is B negative, I have surgery and I need blood. Who can give me blood? B can give blood to B, O can give blood to B, and only negative can give blood to negative. So if I'm B negative, I can receive blood from only B negative and O negative. This is how these diagrams work. Instead of memorizing the donation and receiving, you only need to know the charts. And these charts can help you to easily identify these donations and receiving. There is a disease which we call it hemolytic disease of newborn. When does it happen? If we have Rh negative mother and Rh positive father and they want to have baby, look at this happening. When the father is positive and mother is negative, the Rh of the baby become positive due to Mendelian law. Between fetus and mother, we have placenta. During the last trimester of pregnancy and at the time of birth, sometimes in the site of placenta, the blood of mother and fetus mix a little bit together, and the Rh positive red blood cell come to the body of mother who is negative. In the first pregnancy, nothing happened, but the body of mother start to make antibody against Rh positive, and these antibodies are created in the body of mother. In the second pregnancy with positive baby, this anti-Rh can come to the body of fetus and fight with the RBC of the fetus. So hemolysis and destruction of red blood cell can take place. So if you are Rh negative and you want to become mother, don't worry. We have the solution for that. We have the injection, which we call it ROGAM injection. We can usually administer this injection on week 28 and 72 hours before delivery. It prevents making antibody against Rh and nothing happened to the fetus. And you need to repeat it in the next pregnancies as well. So if you are negative and your husband is positive and you want to have babies, you need to talk to your gynecologist and he or she will administer these ROGAM injections for you. In the last part of this PowerPoint, you have some questions. I want you to do these questions yourself, and on the day of practical, I will go over them with you. But I will do the first one. You have a slide. You have 3 droplets of blood here. One of these droplets is treated with anti-A, one of them is treated with anti-B, and one of them is treated with anti-RH. And you can see agglutination only with anti-A. When you have agglutination with anti-A, only the blood type is A negative. And then I want you to tell me A negative can donate blood to which blood types and can receive blood from which blood types. A negative can donate blood to A negative and positive, AB negative and positive. But A negative can receive blood from A negative and O negative only. You have different examples in this PowerPoint. Please go over the examples and next time we will go over them together.